Thank you, Vishu, for that kind introduction, and also thank you, Bijan, for giving me this opportunity to present. Let me just quickly share and start my presentation. So, I have a chemical engineering background, but I've been working in systems biology, and that is what motivated me to develop some of these platforms where multi data sets can be brought together, which obviously appears very complex and unconnected, but when you look deeper, you see connections that we establish when right kind of knowledge base and data are brought together. So with that in mind, we developed a platform which is called as Investigative Physiology, which sort of brings in different data sets so that it can be applied both as a R&D tool, hypothesis generator, you may call it, and also in personalized precision health. So our motivation was to essentially try to leverage the vast amount of biomedical data and knowledge base that exists in the biopharma R&D and precision healthcare. So as many of you may be aware, in biopharma, there are a lot of R&D challenges in terms of slow therapeutic development, lengthy processes, in bringing delays, new therapies, high failure rates, which many of them not making it to market. There are regulatory complexities that come in, especially if you are, want to build it on natural-based drugs. Data silos, a lot of data available, but they may be in disconnected data sets, which sort of limits discoveries. And increasing demand for precision medicine and how we can subgroup the population and then try to be as precise as possible that is required for a particular individual. Adding to this is the challenges for healthcare delivery as well. There's a lack of precision, a lot of general diagnostic principles are applied and then they tend to fail. Fragmented data for an individual, you may not have across systems data. No real-time insights, so lack of tools for quick and accurate insights. And especially in India, the healthcare delivery, that's a heavy workload, right? Too many patients, in some cases, not right amount of data available, in some cases data, which is available but not used, which is underused. Although these challenges exist, obviously the knowledge base and the biomedical data is the key, and optimal use of this is the key. Therefore, accessibility, quality, integration, and utilization becomes very key. So at MetFlux, we are trying to create an ecosystem for data harmonization, physiology-driven AI insights, and digital tools so that one can, optimally use, integrate, be aware of the quality of the interpretations and access, so that this can be used both in the biopharma R&D space and also by clinicians for personalized healthcare delivery. So with this in mind, that we want an improved accessibility, quality, integration, and strategic utilization, we've been trying to provide, bring in these through digital framework. Trying to bring in a lot of vast amount of data, which are with quality into a repositories, so that interoperable platforms and accessibility is there. Try to bring in rigorous quality controls and standardization for the data that we benchmark, and also the interconnections of these data sets to bring knowledge graphs. Through data integration, through these knowledge graphs, provide advanced tools and methodologies to interpret and analyze on these data sets and knowledge graphs, and have strategic utilization, whether it is R&D or whether you want a formulation or you want to bring out personalized healthcare. How can I bring all this information together? Okay. So our basis is built on the core biological research, right, where omics data plays a very important role along with clinical data sets. So be it genomics, transcriptome, proteomics, metabolomics, or microbiome, We try to silos them in different forms which are accessible, including preclinical and clinical data sets for a particular pathophysiology, be it in vitro data sets or animal models or other intervention studies, including PK/PD kind of analysis and then clinical outcomes that may be there. And at certain level, epidemiological informations and health and disease trends, those can also be aligned into the data sets where different kinds of data, biomarkers, disease definitions, right, and triggers are brought together with validated connections, right? So if ideally, if this were to be possible and scalable, then it has got a true potential of unlocking a lot of insights in terms of a pathophysiology. So with this in mind, we started integrating all these data sets on a platform to bring several of these nodes and variables together and the connections. So the first step is, we build standardized repositories. With that, bring in connections through knowledge graphs, where a lot of AI, ML, and LLMs we use to scale, and then a team of specialists validating them at different levels. And bring in quantitative data as well, over and above the qualitative knowledge graphs that we have, bring in quantitative data as well. So that is where omics data sets become, important, and then build analytical tools around it. So, expert systems analysis, and then systems biology models of very key nodes that are plays an important role. So make the data standardized and put it in proper repositories. Bring in linkages so that knowledge graphs are developed. Incorporate quantitative data where we have confidence in several of these data, and then build analytical tools around it with two applications in mind. So you have therapeutic research, where we try to understand disease mechanisms and pathways, try to identify novel therapeutic targets, optimize treatment strategies, develop risk-tolerant clinical trial design, and forecast real-world treatment outcomes. So this is more in the R&D space. And then also in the precision health management, where we would want take a data from an individual and try to simulate disease progression in these virtual models, and then try to assess disease assessment, and also try to provide the personalized and precise treatment plans, right, using a certain kind of data. And if possible, track and optimize a patient treatment journey, right? So we have been trying to use this framework in both these domains of therapeutic research and precision health management. So during the last 5 years, we have been developing proprietary assets in terms of harmonized multi-source data repository, multi-scale knowledge graphs, which are very disease specific to with validated pathophysiological insights, and build analytical tools on them, right from just connections and graph theoretical analysis to expert systems to dynamic models. And on this evolve platforms where people can access and use them with AI-driven interface and dashboards, automated analytical pipelines. People can query and do analysis on these platforms, and flexible deployment where accessibility is there. So these are getting now scaled and under development. The end goal is that it has to give advanced capabilities of pathophysiology, network analysis, therapeutic target identification, predictive analytics, and personalization, right? Individual and population stratification, with the end goal of therapeutic research and precision health, where biopharma, health management industries, the companies which are developing, have access to a lot of data, so they can use these to come up with precise analysis. So at MetFlux, internally we have been developing platforms very specific to pathophysiology involved in disorders of metabolic aging, disorders of immune health, and the gut-linked diseases, right? So we have built a lot of platforms around that. Child growth and development nutrition, along with very specific to Indian datasets, Indian child datasets, and disorders of skin, right? So these are some of the platforms that with our own engagement with our partners and internally we have been able to develop. So what I will do is I'll take some few examples and then see how all these things come together and look at specifically metabolomics data, given that we are trying to focus more on the metabolomics data sets. So such a knowledge base and graph on which the base interconnections are got, on that we lay the omics data. So for example, I have an expert system on which I would want to know how the metabolomics, okay, the various metabolites which they move out of their homeostasis, how they get integrated into our connections. So we try to build those connections very specifically with validated physiological insights that various studies have brought in. So we integrate that into the knowledge base and then develop algorithms to incorporate these metabolomics data to give very precise outputs. Okay, so our analysis of metabolomics starts post the fold change or the changes that you observe in very key metabolites, right? So it is the post analysis of a metabolomics platform where we want to get insights of that data, right? So where the metabolomics data is used for further analysis, that is where our platform starts, because the other domain of how to get raw data, how to have more accuracies, and the analysis of fold change. So that is a different domain, right? So our starts post that, how we use that data. So using these, individualized and population kind of analysis, pathway analysis, and genetic and personalized suggestion for individual is where we start, interpreting things using such an expert system. So one example that I will take is for the metabolic syndrome, where we have a robust platform that is built. So we start with lifestyle factors at one end, looking at the various biomarkers which plays a role, and then give out output scores, right? In clinically how the output scores go in. And on this, the omics data is overlaid. So there's a base platform which is validated network on which several of these omics data are layered, and then the analytical tools are built around it. So we have input triggers, we have processes and nodes we align, and then we have very precise clinical outputs. So that which we integrate. And one of the specific sub pathways that I can show would be a network like this, right? So the base is that I will have triggers at one end, and I will have very diagnostic parameters on the at the other end. And now these are connected through various nodes and pathways. Some are measurables and some are may not be measurable, but we do get weightages around that. And on this is where the omics data now is overlaid. So this is a validated data with several studies, and the scientists, or clinicians agree that these are the pathways which get disturbed in different organ sets. So each one is tagged with an organ set, like, whether it is liver, pancreas, or it is muscle, whether it is adipose tissue. So these nodes are targeted and they're interconnected in terms of the plasma levels. So a knowledge graph would look something like this, where I will have triggers at one end, maybe psychological stress, sleep disorders, bringing a HPA axis, it gets perturbed, circadian rhythm gets perturbed, HPA axis gets perturbed, and it has its own consequences of several pathways getting affected, right? Or you have a umami food affecting the polyol pathway, right? And glutamate getting IMP getting perturbed, and then it has a cascading effects or domino effect down the line. Right? So we start and bring these physiological network, and on this is where we are going to overlay the omics data and then do a lot of interpretation, right? And then each connection here will have a weightage which comes from the data analytics that we bring in, the AI weightages that we bring in for each of these connections, with several studies that exist in the public domain, and our own studies adds to this, and then we bring in a weightage so that we can run from left to right, or we can go from right to left. You create a pathology and you go left to right and see what are the weightages, or you start with trigger and you can go from left to right, and then also look at the different nodes. I can go and perturb a node. For example, if a particular metabolite is in excess, I can create that change in that metabolite and see just that metabolite increase, how it may flow through in this network. Okay, so these kinds of analysis I can start doing now once I have this base expert system defined. So specific to metabolomics, so we integrate on such a expert system the effects of various metabolites which are known in the metabolic syndrome. For example, we bring in predictions of these effect of these metabolite and validate the physiological outcomes of those. And then we can do whether the model is able to capture a clinical study that is in a population, the variability that exists in the population, how that plays out on this network can be assessed, and then an analysis on an individual who has a diverse metabolic profile. So this is how we sort of integrate, take this expert system, and on that I can bring in
externalized disorders.
Very, very specific to these outcomes. Yeah. So, anybody has some disturbance. I don't know where it is coming from.
Moni, can you please switch the camera of that person off?
Okay, I try.
Sorry, Professor Venkatesh.
No problem. No problem. Yeah. So for our system, for example, if it is a metabolite analysis that we need to do, so for me, I will have a set of metabolites and their levels and how they are changing. So, for example, I have these 30 metabolites which are known to be, the high, statistical significance. Then I can take these now integrate into the network and then try to give out outputs, physiological outputs. So our base is the expert system that I have, and on that I give weightages for a perturbation on a certain level of metabolites, and then look at for example, just to give an example, once we do these analysis and we have confidence in our predictions in terms of a panel of metabolites, then I can go and do a thought experiment. You're able to hear?
Now we hear you again. Yes.
Yeah. So I can do a thought experiment where I can just look at one effect, okay, one effect of a metabolite, and see what changes that it may bring. Right? So, for example, you have say betaine, right? So it is supposed to drastically get affected in metabolic syndrome, right? And either direct or indirect, the betaine levels going down in the plasma is been associated with negative regulation of apoptosis process, immune effects in terms of IL-1 beta production. So it inhibits fatty acid synthesis. There is an upregulation of GLUT4, bringing out glucose homeostasis, and then there is a downregulation of NF-kappa B, right? So once I integrate this, now I can do an analysis on the network and say how betaine levels, okay, if I alter, how it is going to affect glucose homeostasis, or fat homeostasis dysregulation, pancreatic health, hunger and fatigue, blood pressure, right? So we can now give the outcome readouts in terms of a perturbation of a particular metabolite. And obviously, in a real system, there will be multiple metabolites which will go wrong. So in the network, I'm going to take a node which may be betaine, and I'm going to perturb that and then see how it propagates, forcefully changing a particular metabolite, and then look at changes. So these kinds of experiments now one can do using such a platform. So looking at a study where I have a population, so I can take a healthy population and then look at what are the healthy metabolites. Now alter the metabolites, for example, those 30 metabolites that I was looking at, I can alter it now and create a population and see how the disease manifests, and then compare with actual observed clinical studies, right? So I can go and now benchmark, for example, this particular graph is essentially showing a healthy, the first blue is a healthy one. Orange is when the metabolite is perturbed. What are the readouts that it gives, and then the actual disease phenotypes that is there. And you can see that across the metabolites that we have observed, and we perturbed and take a population of metabolic syndrome, group with a metabolic syndrome, which is already manifested, we can now give a readout in terms of the perturbation that exists in glucose homeostasis, fat metabolism, hormonal change in terms of hunger and fatigue, inflammation, blood pressure, which are all hallmark of metabolic syndrome. Okay, dyslipidemia, hypertension, T2D, obesity. So all these are a manifestation of metabolic syndrome. Now we can correlate and say how the expert system along with these nodes are able to validate, okay, in a clinical study. I can take individuals and then look at how their the expert systems throws out. So it'll actually give me readouts of the several nodes in the expert system, connecting from triggers to the outputs. Say, for example, I can go and say in one individual, BCCA synthesis and secondary bile acid is perturbed more than anything, any other node. Some other individual, you have glucotoxicity, inflammation, and amylin metabolism, which have got perturbed. In another individual, right? So you have gut dysbiosis is the major perturbation with SCFA, lowered, short chain fatty acids have got disturbed because of leaky gut. So these kinds of interpretation now I can start doing on individual, on individuals, right? And then get readouts in terms of a network, how that has got perturbed based on the metabolic panel that I have aligned on the platform, right? So I will have readouts for individuals, how quantitatively it might have got perturbed based on, in this case, I can include the blood parameters as well, or the clinical parameters like glucose, fasting glucose, their HbA1c. Also, I can add on the top of the metabolomics that I have to give more validated pathway perturbations that have happened, and also give some triggers, because I can go from the extreme left. I can give weightages in terms of, for an individual, what might have been the perturbation, right? So is it the abnormal meal time, or a sleep disorder, or a micronutrient deficiency, which sort of is giving rise to those pathways? So I can give a weightage in terms of inputs and then validate, ask this patient, whether these effects are, which are indeed being affected, right? So we can have a integration in terms of the triggers as well. Okay. So it's a sort of a combination of the base expert system and how these nodes of the omics data can be overlaid onto this, and give both at the population level, do an analysis, or at an individual level, try to interpret it. Okay. So using these expert systems, integrating data and the knowledge base and the knowledge graphs, okay, one can look at disease risk assessment, dysregulated pathways, personalized risk, on an individual, and create a risk assessment on a population as well, right? With statistical assessment that we do. So I don't know how much time I have. There is another example on the skin pathophysiology, where we have integrated multi-omics. I will not take much time. I'll just sort of indicate that in this case, the platform would have this is the skin, the plasma metabolite, and gut, because even the gut metabolites and the gut status, immune status, may influence individuals with the skin phenotype. So again, here we start with this is very specific to atopic dermatitis. Again, we use AIML techniques and our own methodology to first build a expert system, right? So from triggers to outputs, right? So you have erythema, dryness, moisture loss as an output, and then you have antibiotic effects, right? Cosmetics affecting, giving allergic reactions. So all those on the left, and then you have the gut and the skin pathways, which are sort of integrated with very specific microorganisms also affecting, because you have gut dysbiosis, and on the skin also the microorganisms getting affected, where the microbiome is also sort of interpreted. And so this is the first step for us, validating this, and how quickly can I build and converge on a validated network of trigger to the outcomes with the nodes. And once these intricate interactions are brought in, linking it from multiple triggers to the clinical outcomes, I overlay, the omics data on this and say which nodes they do get affected. For example, in this case, metabolites, some specific amino acids and your ceramides in the gut and on the skin, how they affect, right? Different nodes. We sort of connect and then give weightages, using the data sets and the knowledge that exists. So we give certain weightages, both upregulation and downregulation weightages we align. And in this particular case, I'm also giving you how the omics data and the RNA-seq data is also interpreted on that. So it's a multi-omics analysis where RNA-seq data and metabolite are overlaid on this network. Okay, so then I can look at antimicrobial deficiencies and how infection levels may go up because of the compromised skin integrity. How the ceramide imbalance is creating, the pathophysiology. The sphingomycin dysregulation is occurring, right, in terms of increasing epidermal barrier permeability, right? So which is coming from the gut and what is getting manifested on the skin. So these kinds of interpretations now I can bring in by giving these notes. Similar approach what I showed for metabolic syndrome, but this is in the atopic dermatitis, very specific to this pathophysiology. So we align this and then look at the omics data. So I'll give a very small network which has come out of this analysis, that how flagellin, IL-13, RNA-seq perturbations that occur, which affect the metabolites. Because of these perturbations at the genomic level, you have changes in the lipid and the metabolite alteration that occurs, and that leads to the network domino effect in the network, giving rise to you have the disease pathophysiology. You have quill effects, itch, the oozing, the moisture, this one oozing because of the atopic dermatitis, a SCORAD clinical score, erythema. So all those interpretations now we can do and sort of integrate the omics data together to interpret and come up with a very precise interaction, a subnetwork, in terms of transcriptomes, how they are altering the metabolite, how they are altering a particular node, giving rise to disease symptoms, right? So in both the examples, we have a knowledge graph, validated nodes, bringing in different data sets and bringing in weightages for these connections, okay? And employing these multi-omics data to interpret and elucidate the domino effect of perturbations, right? So essentially, in very specific to omics data, one can characterize diverse pathophysiology states accounting for the omics data. For example, in metabolic syndrome, we have created a net of 89 clusters with very unique metabolite clusters, which represent 89 different sub-phenotype, you may call it, in the metabolic syndrome, right? Where hypertension and dyslipidemia is very high in one cluster, whereas T2D dominates in one cluster. So different nodes and different metabolites are associated with these 89 clusters. So one can bring in such subgroups. You can benchmark new omics data. If a new omics data there, then I can overlay on this and then try to see the existing knowledge and data, how does with confidence limits. So what is known, okay, what is new that is coming out, and what contradicts, right? So I can start asking such questions.